Hello everyone, I'm George Leonov. In this video I will try to review some recent papers in the field of aging, microbiome, obesity and nutrition. The first publication in Nature is about the epigenetic memory of adipose tissue. Obesity is a risk factor for many diseases, including endocrine, cardiovascular, neurological and oncological diseases. The clinical aim of obesity treatment is to reduce body weight to normal level, which can be achieved through lifestyle change, including diet and increased physical activity, as well as the use of medication and bariatric surgery. A common problem preventing successful treatment is weight regain, or the yo-yo effect. Data from previous studies suggest that possible cause of weight regain is associated with epigenetic modification, affecting adipose tissue cells in obese or overweight individuals. However, the molecular mechanisms of this effect remain unclear. The aim of this study was to investigate whether epigenetic change characteristic of obesity persists after significant weight loss and to identify potential mechanisms associated with this effect. The study was conducted in both human and mice using single-nucleus RNA sequencing and other methods to assess epigenetic change. The results of the study showed that adipose tissue, mainly adipocyte precursor, and endothelial cells, retain a gene expression profile characteristic of obesity even after 2 years and a weight loss of more than 25%. The authors showed that reduced expression of genes associated with metabolism and increased expression of genes related with fibrosis and apoptosis were maintained. A study in mice showed similar results. After weight normalization, some metabolic disorders persisted. The expression of genes associated with inflammation was increased and the activity of metabolic genes was reduced. In addition, when the mice returned to high-fat diet, they gained weight more quickly than mice that had remained on the standard diet all their lives. It can be concluded that the effect of a repeated weight gain, yo-yo effect, may be related to the differential expression of genes characteristic of obesity, which persists due to epigenetic modification. The second article, published in the Journal of Translational Medicine, examines the relationship between the gut microbiome and longevity. Dysbiosis is one of the 12 hallmarks of aging and contributes to many pathological conditions associated with age-related diseases, such as diabetes, hypertension, and Alzheimer's disease. Identify patterns of age-dependent change in the gut microbiome may have clinical implications for both monitoring and modifying gut health to achieve healthy aging. The study used data from multiple national studies in several countries, with a sample size of tens of thousands of people. It used GWAS for genetics and 16S RNA-seq to investigate the microbiome. It was shown that general Acinetobacter and allostapes were associated with longer lifespan, likely mediated through lipid homeostasis and cardiovascular health. In addition, the prevalence of subdoligranulum was positively correlated with parental longevity, with slower biological aging, and negatively correlated with frailty. In contrast, Bacteroides miscelensis was associated with accelerating aging. In addition, the stretch degradation pathway was associated with longevity. Akkermansia multifila was negatively correlated with longevity, but positively linked with features of parental longevity, maternal age of death and cumulative parental age of death. Paracettorella was negatively associated with both longevity and life expectancy. Paracettorella also was positively associated with BMI and type 2 diabetes, suggesting that it may have potential effects of metabolic diseases. Helicobacter pylori was also associated with reduced life expectancy, presumably due to the bacterium involvement in the tricarboxylic acid cycle. Acinetobacter showed a positive link with fiber consumption. Another study found a significant increase in the species in individuals fed a diet high in resistant starch. At the same time, the Bacteroides mesolensis increases mainly with high protein and fat intakes. The next paper focuses on the use of exosomes to reduce the progression of skin aging. Skin aging is an inevitable process caused by the superposition of internal and external factors that reduce the structure integrity of the skin and its physiological function. Skin aging is associated with the dysregulation of skin cells and the loss, fragmentation, or fragility of extracellular matrix fibers, which manifest as wrinkles, sagging, and pigmentation disorders. Advanced signs of aging include genomic instability, telomere attrition, epigenetic changes, loss of proteostasis, impaired microautophagy, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell depletion, altered intercellular communication, chronic inflammation, and dysbiosis. Emerging treatments like human platelet extract, rich in platelet-derived exosomes, show promise in improving skin health. Previous study indicated that topical platelet-derived exosomes reduced photo damage signs. This study evaluated its effect on cellular senescence markers and telomere-associated photo after 12 weeks of use. Essentially, the human platelet extract should be similar in the composition to platelet-rich plasma. In this case, the extract is allogenic and standardized, as author point out, unlike PRP. Topical human platelet extract significantly reduced high senescent cells in the dermis. There was also a decrease in telomere damage after topical application. In patients with high senescent cells at baseline, there was a 40% reduction in pro-inflammatory senescent associated secretory phenotype. Overall, there was a decrease in the number of telomere-associated foci in each cell nucleus after treatment. Extracellular matrix remodeling pathways, including collagen and elastic fibers, were upregulated. Paper 4 investigated the relationship between nut consumption and disability-free survival in a cohort of adults greater to 70 years of age. Population aging is a global phenomenon linked with physical and cognitive decline due to factors such as loss of muscle and bone mass, fat redistribution, and oxidative stress. This change increases the risk of falls, fractures, chronic diseases, and dementia. Daily nut consumption was significantly associated with a 23% lower risk of reaching the disability-free survival compared to non-consumers. Previous studies corroborate these findings, showing that high nut consumption is linked to reduced risk of all-cause cardiovascular and cancer mortality, particularly in older population. This result highlights the potential of daily nut consumption to support healthy aging and longevity. For adults aged 70 years and over with suboptimal diets, daily nut consumption is associated with the promotion of healthy lifespan. The latest article focuses on epigenetic markers of aging in Olympic athletes. Over 10,000 athletes competed in the 2024 Paris Summer Olympics, trained up to 10 hours daily. While this intense physical activity carries health risks like cardiac and orthopedic overuse, it also offers significant longevity benefits. Studies show elite athletes often outlive the general population, as seen in Tour de France cyclists and runners who broke the 4-minute mile. Researchers analyzed the DNA methylation profiles of 59 Olympic gold medalists and 332 non-athletes. Using DNA methylation-based aging clock like Grim age and skin blood, they measure biological aging and compared epigenetic markers linked to health and aging. Olympic champions showed slow biological aging compared to non-athletes. Both male and female champions had longer DNA methylation predicted telomeres, a marker of cellular longevity. Male wrestlers exhibited faster biological aging compared to gymnasts, fencers, and water polo players. Female athletes showed no significant differences between sports. Differences in methylation we found in genes related to interleukin-2, STAT5, and mTORC1 signaling pathway, both associated with cell growth and immune function. The results suggest that intense physical activity, even from a young age, does not negatively impact epigenetic aging. Instead, elite athletes demonstrate a slower biological aging process, contributing to reduced mortality and lower disease risk. In 2020, virologist Bita Halassi faced a devastating diagnosis, a second recurrence of breast cancer in the site of the previous mastectomy. Inviting to undergo another round of chemotherapy, she decided to take an extraordinary step, turning to her expertise in virology and experiment on herself. Drawing to her knowledge of cultivating viruses, Halassi developed a treatment called oncolytic virotherapy, an imaging approach that used viruses to target and to destroy cancer cells by activating the immune system to fight the diseases. Halassi chose two specific viruses for her treatment: a measles virus commonly used to childhood vaccines, and vesicular stomatitis virus, known to cause only mild flu-like symptoms. Over a 2-month period, she injected these lab-grown viruses directly into her tumor, working under the supervision of her oncologist to ensure she could switch to a conventional treatment if necessary. The results were extraordinary. Her tumor shrunk, softened, and separated from the invasive tissue it had grown into, making it easy to remove surgically. Analysis of the tumor revealed a robust immune system with a lymphocyte infiltration the cancerous tissue, a clear sign that the therapy had provoked her immune system into action. Following surgery, Halassi completed a year-long course of the anti-cancer drug trastuzumab, and she has now been cancer-free for 4 years. The therapy remains experimental, with only one virus-based treatment approved for metastatic melanoma and none for breast cancer. Nevertheless, Halassi's case highlights the untapped potential of using viruses to treat early-stage cancer.